Okay, our next presenter is Linda Marbán from Capricor.
Good afternoon. Please excuse my voice. I am recovering from laryngitis, and if I have a coughing fit, don't send in the rescue crew. I'll be okay in a few minutes. My name is Linda Marbán, as I was just introduced, and I'm here to talk to you today about Capricor. We have a cell therapy product for heart disease that has taken us to a clinical stage company. We just started recruiting our phase 1/2 clinical trial this week. So the disease that we're targeting to treat is heart failure, which is seen at the far right end of the diagram you see at the top there. But we're targeting it early on in the development of the disease and trying to treat heart attacks. So just let me walk you through very quickly, although most of you have heard this probably 20 or 30 times today, exactly what happens when you have a heart attack. Blood flow is stopped to the vessel of attack. Usually it's the left anterior descending artery, which leads to tissue dying. The injury essentially spreads, so more cells die over time, short period of time that leads to what we would call the scar or infarct size. And so you'll hear me talk about that over the next little bit and why we think that's an important parameter to be addressing in this clinical paradigm. And then ultimately, that tissue thins. It's no longer muscle tissue, it's scar tissue. The heart has to beat against that load that's no longer helping it, which ultimately leads to the development of heart failure when the heart can no longer compensate for the increased demand without the present muscle to meet the needs of the body. The therapy that we have that you're going to be hearing about aims to attenuate that remodeling process that is so well known and has been the subject of attack for many, many years in the cardiac therapeutic arena. To date, cell therapy has been tried in the heart with using many different cell types, and you've heard about them: fat cells, muscle cells, bone cells, all kinds of cells. We have a cardiac cell. It's a cell that comes from the heart. It's a mix of different cell types that we believe and how have data to show are the very best at treating the heart. Why wouldn't a cell from the heart be the best to treat the heart? There has been no data shown to date that shows a reduction of the scar, regeneration of new cardiac tissue, and the corresponding improvement of function that we also desire to see in patients with heart disease. Capricor was founded in 2005 in Baltimore. It's a spin-off of technology from the Johns Hopkins University, the University of Rome, and now Cedars-Sinai Medical Center in Los Angeles, where the company is now based. We've been able to get to this important milestone of a phase 1/2 trial by taking in very, very low equity investments, only $12 million to date, and that's through angel funding, because we've been able to leverage our academic partnerships, both with the labs of Eduardo Marbán at Cedars-Sinai Medical Center, and also through the luck and skill of our own organization to achieve grants, which fund our R&D programs. To date, we have a disease team grant from CIRM and several NIH grants, including, I have to update the slide, a new one that was just awarded a few days ago. We have two products under development, the CardioCEUS and the cardiac-derived cell, which I'll talk more about in a moment. And as I mentioned, our intellectual portfolio is growing based on original intellectual property from University of Rome and our colleagues Elisa Messino and Alessandro Giacomello, and then followed up at Johns Hopkins University by the revolutionary work of Rachel Smith in the audience and her mentor, Eduardo Marbán. And then the work has been developed further at Cedars-Sinai Medical Center with new intellectual property both from the hospital and from Capricor itself. So Capricor is in a good position financially. We have $5 million in the bank and no debt, which is a nice thing for a company of our size, and we're funded completely through our phase 2 milestone. The phase 1 is funded by a Recovery Act grant from the NIH, and then the phase 2 will be funded through the disease team grant of CIRM, which is really nice to be standing in my shoes today, because I can focus my energies and efforts in being able to make sure that the trials that we do are the very best and that the data that we generate changes the industry, as opposed to being out there and worrying about how we're going to fund our trials. So as I mentioned a few moments ago, we have two product candidates, the cardiac-derived cell and the cardiosphere. And shown in the simple diagrammatic representation above, you'll see our manufacturing process, which I'll share with you more about in a few moments. But the cardiosphere-derived cell is shown in the far right, and I think this is the pointer. Yep, shown right here. And it's a single cell monolayer that can be injected nicely down a coronary artery, and that's what we're focusing on right now, is this very simple procedure done in every cath lab across the world, where you can quickly inject the cells down the artery and send the patients home. And we're focusing our efforts right now in what we call a convalescent MI population. Those are patients that have had a heart attack within the last 1 year. So how do our cells work? And it's interesting, when I was thinking about this talk, I think work is an interesting word. You make your slides and you give talks many times, you don't think a lot about your verbiage. And then I thought, work to do what? What are we really looking to do? And it's interesting, we know what they do, but we don't know really how they work. And that's what we're going to be finding out over the next few years in our clinical trials and development of all the mechanistic assays that we know we need to do for phase 3. What we know they do is that they decrease apoptosis, that is programmed cell death, telling cells that you're not healthy and you need to go away or die. One of the most important things they do, and we now have some beautiful data that's been published recently, that shows that it actually recruits endogenous stem cells. So our cells go in there and they tell the body, tell the heart that something's not right, and they bring in all of their friends that are supposed to be there and help to make new muscle. Way more muscle is created, as you'll see in a few moments, than we would anticipate based on the number of cells we inject, and also the fact that they disappear. They stimulate new cardiomyocyte proliferation. We know that there's actually new myocardium that's created. And they improve blood vessel density. They're angiogenic, which is a great thing in an organ that's now been starved for oxygen and other blood-delivered products. We think of our CDCs as a local drug delivery system. They're eventually cleared from the heart, but they get in there and they release all kinds of growth factors and paracrine factors and other good things, some of which we know about, some of which we don't, that stimulate the heart to repair the injury that's been done to it, which for those of us who have spent our lives thinking about how to fix a broken heart, it's quite exciting to think that we finally have a way of mending the heart. To date, we are the only therapeutic that both reduces infarct size and promotes regeneration of cardiac muscle. And I'll show you that in a few moments. So I want to take just a moment to talk about our clinical development programs. In a few moments, I'll tell you about CADUCEUS, the clinical trial that was done as a relationship between Cedars-Sinai Medical Center and Johns Hopkins University, also funded by the NIH, that looked at our autologous, taken from you and put back in use CDCs, and was recently published in The Lancet. As we go through the next couple slides, you'll see that as I begin to talk about what Capricor is doing, Capricor took that autologous paradigm, and while Cedars and Hopkins were busily treating patients and wondering what was going to happen, we made a commitment to develop a cell therapy that was also a good business model. And so we developed an allogeneic cell product, and that's what we're moving forward with today. So let me just tell you a little bit about CADUCEUS. CADUCEUS, as I mentioned, conducted by Cedars and Johns Hopkins, studied the intracoronary delivery of the autologous CDCs in patients that had a recent heart attack. And those patients had to have a reduced EF and able to qualify for the trial. Now, why was that patient population chosen 30 to 90 days post-MI? It was very simple, because those patients could then get their cells. So they had to come back in after their heart attack. They needed to have an invasive biopsy. Then that biopsy needed to be carried very carefully or shipped carefully back to our labs at Cedars, where it was grown up into a patient dose. And I ran that manufacturing program along with Dr. Smith. And what we realized very soon into this is that it is a huge nail biter to do autologous cell therapy. Not only do you have all of your processes and quality to control each and every time with a patient having undergone a procedure to give you their tissue, but there's factors that are out of your control. Do they have a karyotypic abnormality when they grow? Is there some reason that that patient doesn't achieve dose? And so we became highly motivated, even though this trial was extraordinarily successful, to switch to an allogeneic product and do that validation work, which we did during the trial. Let me just share with you the very exciting data. I don't want to take anything away from CADUCEUS, although I think somebody said earlier that your phase 1 data is forgotten pretty much as soon as it's published. I don't actually think that's true. I think that we still get to ride this very exciting wave of the fact that 17 patients received our cells and 17 patients had a reduction in the size of the damage done to their heart. The average was that there was a 50% reduction in the scar with a commensurate increase in new viable myocardium, and it's been demonstrated now not only in the humans that were studied looking at their MRI images, but also in follow-on studies in large animal models that in fact it is new myocardium that was created. So we are very proud of and excited by this data that shows that our exact cell type, the CDC, leads to a reduction in infarct size. And therefore, what we don't know and what we think should happen based on all of the supporting data in the field is that then our patients should go on to have a longer event-free survival, better quality of life, and less hospitalizations than the untreated patients. So meanwhile, as I mentioned, we were working on the allogeneic paradigm. And of course, the first thing we had to do was show product equivalence, which we ultimately did. The cells, interestingly, are very safe. They lead to no cellular or humoral immune response. We did significant studies with the Harvard Transplant Center and the lab of David Sachs, very well known for his work in understanding rejection and transplant immunology. We saw no systemic inflammation, no permanent engraftment of our cells, which actually, when I started this company 7 years ago, if I said the cells disappeared, I probably would have gone home because I thought, in the day that we wanted to inject the cells, and I envisioned these cells dividing and growing and making brand new heart muscle, and that was, what we thought we were doing. And what we now know is our cells are causing regeneration of new tissue, but in reality, they're not the new tissue themselves. They just go in there and do their work and leave. So there's no permanent engraftment. They're all gone after about 3 weeks. There were no laboratory abnormalities and no related histological findings. We did ISHLT grading, which any of you that knows the transplant field is the gold standard to look for rejection. We didn't see rejection. And they worked the same, some might argue a little bit better, than the autologous cells. They led to a reduction in infarct size, improvements in the ejection fraction in the pigs. There were trends for enhanced contractility, better ability of the heart to eject the blood, and they were completely comparable to the autologous cell. So now we've shifted our focus on allogeneic cells. So we haven't abandoned our autologous roots, and for the right indication, we'll revisit that again because I think there is a continuum of care that we will ultimately develop in cell therapy. But right now, we're developing hard and fast our allogeneic cell. We're recruiting AllStar, our new trial, phase 1/2, allogeneic heart stem cells to achieve myocardial regeneration. You can see sort of the standard laundry list of inclusion criteria shown below. We're going to look for ejection fraction less than 45% by MRI, and MRI is more accurate than echocardiogram, so that's why we're going a little bit higher. But we're also, because our gold standard is infarct size, we're also looking for a certain size of a heart attack. So we actually are doing MRI. We have a great imaging core that will do a 48-hour turnaround for us, and we make sure that people have had a heart attack that's greater than 15% of their myocardium before we accept them into our trial. So therefore, we're not going to miss people with big infarcts and high EFs, or conversely, people with small infarcts but low EFs. We're going to do, at least initially, a stratified patient recruitment so that we make sure that this therapy is safe and doesn't cause immune response, which we don't expect it to. Then we move on to the double-blind, randomized, placebo-controlled portion of this trial. That will be 20 sites, the allogeneic CDCs, so it's standard intracoronary infusion. The endpoints, of course, are safety. That's the most important thing we want to do, but I would be remiss if I didn't say that what we really want to do is see if our cells help people live longer and better lives. And the first step is, of course, making sure they do no harm. The primary safety endpoint is a 1-month endpoint, which is really nice because it allows us to move to phase 2 relatively quickly. It's 14 patients. And then we continue to monitor the safety in the 260 patients that will be followed on for the placebo control point. Our primary efficacy endpoint is a 12-month relative reduction in infarct size, although we will be looking at 6 months, and based on the CADUCEUS data, we expect to achieve both our secondary endpoint of 6 months and our primary of 12 months. So look for data from us in probably end of 2014, beginning of 2015, as we move into our phase 3, although I hope to be back here before then. And we're recruiting patients now for the phase 1. So I'll just briefly talk about our manufacturing program. I'm very proud of it. It's something that we've done in-house. We built our own facility too. We always have our eye on the ball in terms of getting through BLA into the market, and so we want to be a step ahead. And let me just walk you through very quickly why we think the allogeneic cell is our best cell and how we're growing them up. It's an off-the-shelf availability product. We're working on all kinds of things so that the shelf life permits usage for a long time and that there are doses always ready. And I think people have touted proudly today, and we are up there touting proudly as well, that pull a bag from the freezer and run it down to the cath lab and your patient is treated within an hour. There's no invasive procedures for your patients necessary. It's reproducible, and the best part for me is QA, QC, and manufacturing. And if there's a manufacturing failure, it's a headache for us and the home team, but no patient is going to suffer for it, which is terrific, and reassures us a lot that we're giving the patients the very, very best that they expect from us and that, of course, we want to give them. The process failures are not linked to the patient treatment. Like I said, we go home and beat ourselves on the head, but they know nothing. And the cost is significantly reduced, which is nice. It's a tenfold reduction in cost, and that's mostly because of manpower issues. Shown here is a schematic of our manufacturing paradigm. We have relationships with 2 local organ procurement organizations in Southern California, and ultimately we'll increase that over time. We bring in hearts that are of transplant quality, but for some reason have not been able to find a home, and so we make good use of making thousands of doses of cells from those hearts. There's a special process that's been developed by our team to dissect the heart tissue and then put it into cell culture, where the outgrowth shown here, this is called our EDC product, and you can see the phase bright cells coming off of the tissue in the center. It makes our master cell bank. And then those are ultimately frozen down and tested for all kinds of nasty viruses and things that could happen. And then when we need patient doses, and we usually make those in sort of a 3-month supply right now, the master cell bank is taken out of the freezer, and we grow our cardiospheres in suspension culture. And cardiospheres have been fondly called little hearts in a dish. They're spherical and 3D microtissue product with different cells on the inside and the outside. And then they're too big to inject down a coronary artery. And again, there have been jewels said here today. Noga delivery seemed so sexy when we started this project 5 or 6 years ago, and now it just seems like a laborious, time-consuming process that we'd rather go down a coronary artery. So if you think of a cardiosphere in suspension as a melting snowball, as I do, we plate it, they melt, and all the good cells that were on the inside as well as those on the outside are in this monolayer culture, and then we gather those up, and now they're not in vials, they're in bags, and they can get to whoever needs them to make them better and reduce the size of the damage to their heart. So in summary, and I appreciate your attention at the end of a long day, especially talking about another product for heart disease. We are pioneering the use of cardiac stem cells to treat heart attack patients and ultimately stop the progression to heart failure. We are the only therapeutic cell that has been demonstrated to induce both a relative reduction in infarct size and a regeneration of heart tissue. And we can reduce the outcomes for heart attack patients in the development of heart failure. This has a large market opportunity. I actually almost never present a market opportunity slide because it's just so vast that I think everybody knows that if we could reduce hospitalizations, reduce the cost of healthcare dollars spent on heart disease, we would have a slam dunk product here. And so we really focus hard on the fact that we might be able to transform the lives of people and help them live longer and better. And we had a patient come with us to Sacramento or San Francisco a few months ago to talk to CERM, and he was a patient in the CADUCEUS trial, and he really believes that his life has been changed by these cells, that he will never stop being grateful for the opportunity to be in this clinical trial. We hope we have many more people like that. We couldn't do the great work that we do without the most incredible team, highlighted by Rachel Smith, who runs our R&D department, and manufacturing facility designed and built and cared for by Michelle Creek, and our clinical operations run very capably by Francie, who comes to us through Astrum and Pfizer. And the rest are just the people back home doing the hard work while I get to go and talk to you. I'd like to close with the CADUCEUS data. I think this is a slide that's worth closing with. It shows that the reduction of infarct size shown in the left is very real and very large, with a commensurate increase in viable new myocardium, and that is what we hope to replicate in AllStar. So watch for the data. Thank you very much.